I was really sort of gratified to sort of get an email from Sung Ki sort of after we were sort of making plans for this symposium, for my attendance to this symposium, telling me that he had sort of become the CEO of IPK. And I just think that this is a fantastic sort of leadership choice. I have admired his work in hepatitis C and in virology for decades, and I think his reign here at IPK is just going to be a fantastic success. So congratulations on this position. So I'm going to talk a little bit about sort of what we've been up to sort of lately with hepatitis B and hepatitis C, but I think sort of all of you sort of know one of the reasons that we're interested in these hepatitis viruses, and that is that chronic infections are associated with the development of liver cancer. And sort of liver cancer is, in terms of new infections on an annual basis, it comes in at number 6. And in terms of sort of global annual deaths, it's even higher up in the hierarchy here, number 2. So this is really a huge problem. And the sort of precipitating factors associated with the development of liver cancer, primarily hepatocellular carcinoma, or HCC, have to do with, basically sort of 3 sort of major etiologies. Alcohol, which is a problem in the US and other countries, chronic hepatitis B and chronic hepatitis C, and you can see in this terms of this global map that the sort of associated causes really do vary by sort of country and society. So I think this is a sort of a major goal of really trying to protect against infection or cure infection to actually minimize the risk of developing this more severe pathology, cancer or cirrhosis in particular. So, confronted with this, I'm a molecular virologist, not necessarily a cancer biologist, but I guess what you can do is, sort of prevent infection. So we have, a good vaccine for hepatitis B and HPV, a papillomavirus. And we would like to sort of eliminate the virus, develop curative therapies. We have that for sort of HCV, so that's good news. But I think there are still many challenges that remain in terms of really addressing these problems on a global scale. So I'm going to cover 2 things, and I thought I would talk about some relatively new work in the lab rather than sort of going through sort of a historical sort of journey through sort of the history of hepatitis C. And we really are newcomers to working on hepatitis B. There are probably people in the audience that have been working on this virus for decades. And I'll sort of talk about some of our new work on hepatitis B, and then also for hepatitis C, a model that we're very excited about that may be able to provide some understanding of how chronic hepatitis C infection actually leads to hepatocellular carcinoma. So quite a bit to cover. So in terms of HPV, I think you all know, sort of what the history of this is. After the discovery of the virus, there was a prophylactic vaccine approved in 1981, actually sort of using material from sort of chronically infected individuals. That was later changed to a recombinant antigen vaccine. There are further improvements. There is effective chemotherapy. I'll say a little bit more about that sort of later, but certainly the sort of the nukes that we have now for treating hepatitis B are very effective, but you have to be under treatment, and they don't actually sort of cure the virus infection like we can do with hepatitis C. I think with these advances, people tended to say, well, sort of the problem's behind us. And so research was deprioritized for hepatitis B, and I think sort of shifted more towards hepatitis C after the discovery of that virus in 1989. And we still have millions of people that are chronically infected today that are not being treated with these drugs. And so I think there has been a reemergence of interest in working on hepatitis B. So it's it's come back to a certain extent, and I think people are trying to come up with what we would call a functional cure, because one of the problems with the drugs that suppress the virus is that they don't actually cure infection. So if you stop taking your drugs, the virus comes back, a la, sort of HIV. So in terms of, I call this sort of HBV situation a pandemic. I mean, this is something that we know is a problem. And yet, sort of we still have a lot of work to do. And there are an estimated 300 million people that are chronically infected with hepatitis B in the world. And, there are more than 1 million new infections a year, almost 1 million deaths per year due to hepatitis B. And we have effective vaccines, as I've mentioned, but there's relatively poor global uptake. I mean, some countries have been, sort of fantastic, others have not. And the treatments, as I mentioned, are not curative. And of all of those people that are chronically infected, I think the estimates are that only about 5% of them are actually being treated. So the rest of them are still at risk. So I think, as I said, there's new optimism given the successes in HCV to really tackle HBV. That's fairly widespread, although I think in infectious disease we kind of see this sort of cyclic thing of sort of research support. You would think after a pandemic like SARS-CoV-2 that sort of infectious disease research would be prioritized and maintained for a sustained period of time. That doesn't seem to be the case, at least in the US, where I think we've already sort of taken for granted that we're going to be reactive, sort of rather than proactive. Anyway, a number of direct-acting antiviral targets are being pursued for HBV in terms of drug development, capsid assembly modulators, monoclonal antibodies, antisense oligonucleotides. And I think for many of you that have, and I think the IPK has been interested in sort of drug discovery. I mean, if you're trying to prioritize compounds that have the sort of anti-pathogen effect, you would actually like to know something about what the resistance profile of your virus in the case of HBV is going to be. Is it a low barrier to resistance? If you're trying to choose, amongst, say, 6 compounds that you're going to take forward into much more expensive preclinical development, then, sort of which ones are you going to pick? And if one of them really has a high barrier to resistance and another one doesn't, that might sort of influence what you choose. So I think that's very important, and that's something that we don't have for HBV because this virus really doesn't replicate that well in cell culture. So it's not like you can just take your compound and select for resistant variants and sort of characterize their fitness. In the case of resistance mutations in HBV, we found out about these often in the context of actually having an approved drug that gets used in the clinic and then resistance emerges. So this is one of the things that we thought maybe we could sort of have an impact on, and as I said, we're relatively newcomers to working on hepatitis B, and I think we were naive. This, relative to hepatitis C, hepatitis B is really a complicated little virus. I had sort of no idea sort of how many complexities this virus has. And the difficulty with studying it, it's 3.2 kb genome. You'd think, sort of how much trouble can a little genome like that cause? Well, I've told you that quite a bit of trouble. And, how does it manage to do that? It has all these overlapping reading frames. People haven't been able to make, sort of convenient reporter viruses that really sort of retain all of the sort of viral properties that you would like to have. So the people in my lab that started working on this were really getting, sort of pretty frustrated. And so let me tell you a little bit about the life cycle. I won't go through this in any detail, but this is an enveloped sort of DNA virus. It has this relaxed circular genome RNA. It sort of enters the cell, sort of delivers the capsid. That capsid is uncoded, and then the relaxed circular DNA is transported, is released into the nucleus, and then that gets repaired to produce what is called cccDNA, covalently closed circular DNA. And this is this very stable troublemaker that just hangs around that the antiviral drugs really don't get rid of. It's also the template for transcription of all of the HBV sort of messenger RNAs. So, and that is done by host polymerase II, and the sort of 6 RNAs that are shown here. And then the pregenomic RNA, which basically is the sort of precursor to making new sort of DNA genomes, is translated. And when that's translated, the sort of polymerase that's encoded by that pregenomic RNA actually gets packaged into a capsid, and it's that packaged capsid that sort of triggers the sort of reverse transcription reaction that leads to this relaxed circular DNA genome. So as I mentioned, I point out one more thing, that actually assembled capsid then can, sort of be packaged with an envelope and released from the cell, or it can actually sort of go back into the nucleus using this sort of recycling pathway and be a precursor to making more cccDNA. So as I mentioned, the problem studying this virus is that if you have a cell culture model, which we do for hepatitis B, it often takes hundreds of particles of the virus in order to initiate a productive infection. So you have this very high background of input DNA. And that's true when you actually launch the sort of viral replication using a plasmid, 1.3x plasmid. So again, you have this very high DNA background. If you look at a marker of sort of HBV protein expression and you sort of compare what you see with a mutant where the reverse transcriptase polymerase is dead, you can see here that if you look at the level of core antigen or at the level of surface antigen, there's really no difference between sort of having a replication incompetent sort of way of delivering the genome versus a wild type. And that's also true at the DNA level, unless you do sort of more extensive analysis to actually sort of eliminate the carryover DNA and sort of restrict your analysis to encapsulated sort of DNA. So we got to thinking about, well, how could we get around this problem? And sort of came up with a strategy to think about, could we actually sort of launch HBV replication using RNA instead of DNA? And that would allow us to basically sort of eliminate this background of DNA and sort of potentially do things sort of that we were interested in doing that wouldn't be possible with the sort of conventional ways of studying this virus. And the pregenomic RNA sort of was the obvious candidate to try this. And so you can actually make a plasmid and use that as a template to transcribe unlimited quantities of this pregenomic RNA and then transfect it into cells and see what happens. And it turned out that we thought of doing this for HBV, and it does work, as I'll show you in a moment. But sort of going back into the literature, it actually turned out that Jesse Summers, who's one of the really sort of fantastic researchers in the HBV field, had actually done this for duck hepatitis virus something like sort of 30 years earlier. Anyway, it actually does work. So if you take this transcribed pregenomic RNA, transfect it into permissive cells, you can get reverse transcription occurring to produce relaxed circular DNA. And this just gives you an idea of what the signal-to-noise ratio is in this assay, sort of compared to sort of what I showed you. So if you have a wild-type pregenomic RNA, or one which actually has a mutation that inactivates the reverse transcriptase, it's like 4 or 5, sort of orders of magnitude difference. So actually a very clean system. We could also show that this produces covalently closed circular sort of DNA because, and again, I won't show you all the assays, but you can see the production of surface antigen, which, as I mentioned, has to be made by transcription of cccDNA. It turns out that we do have a complete life cycle. This doesn't actually produce a lot of material in terms of infectious virus production, but if you transfect primary human hepatocytes with pregenomic RNA and then engraft those transfected hepatocytes into a humanized mouse, and these are models that we use a lot these days, which basically have an compromised mouse that allows engraftment of human hepatocytes and expansion in the mouse liver, where you can actually get animals that have like 90% human hepatocytes. And if you inject these pregenomic RNA transfected hepatocytes in, you can actually see that these animals become infected with hepatitis B, and you can see an increasing titer here in several of these engrafted animals. So it works for sort of the HCV genotypes. I think we've looked at 8 of them now. This just sort of shows you what we see, not using a polymerase dead control, but in treating with entecavir to sort of block the reverse transcriptase activity. It works for the major genotypes. There are actually 10 genotypes of hepatitis B, so it's actually quite a diverse virus. And then in an immunoassay looking at surface antigen, you can see in the lower right that we see sort of production for all of these except for genotype G, which is actually very bad at sort of secreting surface antigen, and we think that's the reason why we don't see much of a signal there. So what could we use this for in terms of predicting drug resistance? So if you have sort of a diverse collection of pregenomic RNAs transfected into a hepatoma cell that we use to sort of do these experiments, in the presence or absence of drug, let reverse transcription occur, and if you basically sequence the input and sequence the output, you can look and see basically what sort of mutations are sort of enriched in the presence of the drug relative to being in the absence of the drug. And that actually allows you to sort of map, basically in a single step, sort of resistance mutations to, in this case, polymerase inhibitors. So in the case of T7, it turns out that, you know, like all sort of DNA-dependent RNA polymerases, this makes a mistake. And the error rate is about 10 to the minus 4. So whenever you make an RNA population that you've transcribed in vitro, you're actually, it's not sort of the same sequence as the plasmid template that you use to make it. It's actually quite heterogeneous. So you can use that population to do this experiment, and we did it in the presence or absence of lamivudine, which was one of the first drugs approved to treat hepatitis B, which arguably has a low barrier to resistance. And then basically we could amplify sort of the region, including the sort of polymerase region, and actually enriched were actually the 2 most common mutations that are seen in the clinic. Excuse me, showing up here, sort of methionine 204 to valine or isoleucine. But there are also other sort of resistance mutations that have shown up in the clinic that we didn't pick up here. And so it turns out that, you know, T7 polymerase on the average is going to just make, you know, 1 base change in a given sort of pregenomic RNA. And, you know, going from a methionine, say, at this position, at position 204, there's really sort of a limited number of single nucleotide substitutions that are going to give rise to amino acid changes. And you can see here there are relatively few of these. But these days, and this is way different than when I was a student and you could just make, you know, sort of 1 mutant and characterize it, and that was a PhD thesis. You can really use basically these libraries, deep mutational scanning libraries, to change every codon to all other possible codons and interrogate these guys all at the same time. Really amazing. So we did that and sort of made a library in the reverse transcriptase and did this analysis. And this just sort of shows you what the differences that you could see. And so this is, in blue, is an AlphaFold model of the HBB polymerase, sort of focusing on the reverse transcriptase sort of domain. Modeled into that is some double-stranded DNA shown here in yellow. And then the sort of the drug resistance mutations, the clinical ones, are these sort of red balls that are shown here. And then as you'll see, there are a whole bunch of sort of lavender pink balls that have actually not been seen for HBV. And the question is, you know, sort of why is that? And it's really quite interesting because some of these sort of resistance mutations that are showing up here that haven't been selected for HBV were in fact seen when this drug was used to treat HIV. Excuse me, I've got kind of a scratchy throat here today. And I think one of the reasons for that is that, as I said before, this viral genome basically has all of these overlapping reading frames. And this region of the reverse transcriptase actually overlaps sort of with the surface antigen open reading frame. So you can imagine that, sort of mutations that could confer resistance to this nucleotide are actually going to be selected against because they're actually changing a key part of the surface antigen coding sequence. So, excuse me. This just sort of shows you what we can actually analyze using this RNA launch system. So I've shown you what the sort of signal-to-noise ratio is from a wild-type pregenomic RNA to one that has a dead polymerase. If you introduce a stop codon into the core, which actually overlaps with the polymerase, you can see that that's absolutely essential, whereas the surface antigen, since we're actually not interrogating that part of the life cycle, basically is not affected. So you can introduce in-frame codons in the surface antigen and the HBX open reading frame, really sort of very little to no effect on the ability of this pregenomic RNA to make DNA. So, what about this overlapping region with the core? This is, again, another overlapping region. And so let me sort of orient you a bit on the sort of deep mutational scanning data. So if you look on the right, this is sort of the first 15 amino acids or so of the polymerase reading frame. And if it's dark blue or blue, that basically means that it's sort of deselected. So that's bad. If it's white, that means it's neutral. And if it's red, you can see very little evidence of things that actually make DNA synthesis better. So, you can see that if you look at the polymerase, methionine is really important to have to sort of initiate the sort of polymerase. And several other residues are, sort of very important, and certain substitutions are bad. If you look at the core, and this is from a non-overlapping region of the core, you can see that even stop codons don't appear to be selected against. And sort of why is that? And the reason for that is that this method, when we're sort of transfecting cells with a collection of different variant RNAs, that basically the core can actually function in trans. So wild-type core can actually package genomes that are defective in core production. So, excuse me. I think I must have caught something at the KSBMB meeting. Anyway, I'm sure those kinds of things don't happen here at IPK. Anyway, so yeah, the sort of core can function in trans, but actually the polymerase likes to function in cis. So, basically if you have a polymerase that's not happy, it basically isn't able to sort of copy its encoding sort of RNA, sort of package it into a capsid and be registered here. So if you expand this deep mutational scanning to the entire sort of polymerase, you can see some fairly striking things. I mean, I pointed out the methionine. If you look at stop codons, those are very strongly selected against. That's what you would expect if you aren't sort of expressing the open reading frame. There are sort of other things that are notable. If you look at prolines here, prolines sort of when they're put into alpha helices and sort of other things in the structure are very deleterious for protein structure and function. Now we got the hot water. Thank you. And then you can see some other sort of notable things. I don't have a pointer here, but if you sort of look in these areas that don't show blue, I mean, these are areas that are, just not selected against. So one of them is the spacer region, which is basically shown here. And you can basically substitute just about anything in that region. And, the polymerase is perfectly happy. And, that presumably is because this spacer region is really devoted not for polymerase activity, it overlaps with the surface antigen. It's important for surface antigen activity. And there was some beautiful work from Ralph Bartenschlager's lab sort of looking at hepatitis B-related viruses in nature that really sort of suggest that there actually are, sort of these viruses that are called nicadnaviruses. They're non-enveloped, and they don't have a spacer, which kind of makes sense. And the ones that are enveloped have sometime in evolution sort of picked up a sequence that basically, sort of provides, some selective advantage, presumably, ultimately, sort of being enveloped. The spacer then sort of gets, recombined into this region of the genome and evolves, and then actually that sort of extends down into the overlapping reading frame with polymerase to basically select for an expanded region in the surface antigen, which is involved in receptor binding and other important activities. Good. So I want to point out another thing which really has sort of caught our attention, and that is sort of looking down here at sort of the C-terminal region of the polymerase, which basically has these domains, the sort of terminal protein sort of domain, reverse transcriptase domain, and then that's followed by the RNase H sort of domain. And that's shown here in this structural model. Downstream from that, there's actually this tail, which AlphaFold, really can't do much with. So it's sort of one of these things that people call as being unstructured, but that usually means we just haven't figured out the right circumstances in order to structure them. So what was going on there? And we got interested in this because if you look at the last two residues of polymerase, they're both proline residues. And as I told you, prolines are, very specialized amino acids, generally sort of bad for perturbing RNA structure or DNA structure or protein structures. But in the case of these two prolines at the end of polymerase, you can see that basically all four proline codons are tolerated, and very little else is tolerated there. So it just seems strange that this protein would terminate with two proline residues. Well, it turns out that proline residues, particularly diproline residues, actually are involved in ribosome stalling because they have a very hard time sort of hydrolyzing that bond and releasing the polypeptide. And, we sort of discovered this just by, sort of being completely ignorant of this whole process and, just trying to figure out what these two prolines could be doing. And so it turns out that they stall ribosomes, and Ying Pu actually looked at this, and what you're, on this protein gel, you're looking at, basically a in vitro translation reaction with the RNase H domain and then this tail. And, you can see this band that's a little smaller than 37 kilodaltons, which is basically showing up on the sort of the wild type sequence. And if you treat that with ribonuclease, it's actually got covalently linked prolyl transfer RNA. It basically goes away. And if you make a mutant where you've changed those prolines to alanines, which wouldn't be associated with stalling, then you can see you don't really get this stalled product. So it really looked like, this was involved in sort of stalling ribosomes. And, sort of why would it do that? Well, I think one of the amazing things about sort of many viruses is to sort of how they have evolved to really be efficient machines. And the HBV polymerase is really, I mean, for many years, it's actually been known that this, as I mentioned, functions mainly in cis. So if you think about the model of how this works, you've got, basically this messenger RNA that encodes the capsid protein, overlaps sort of with the beginning of pregenomic RNA. And it turns out that sort of the sort of core protein sort of reading frame and sort of really sort of regulates the initiation of translation of the polymerase. So there's a sort of a leaky scanning model such that relative to sort of translation of the core protein, the polymerase initiates translation at a much reduced level, about 200 times less efficiently than the core protein. So there's a vast excess of core protein. The polymerase, eventually gets made from this pregenomic RNA, and then the polymerase actually binds to a packaging signal on the same RNA that made it, sort of drives this encapsulation process with this sort of excess of core. And then, as I mentioned, that assembled, capsid is what triggers reverse transcriptase activity. So really quite interesting. And so we thought that, well, perhaps, sort of this diproline was actually important for sort of regulating the folding of the upstream polymerase so that it could actually fold properly, interact with the cis-acting sort of packaging signal, and lead to encapsulation. So Mengyang Zhang sort of actually tackled this problem, and I'll just sort of walk you through what she did. So the experiment is to basically take sort of wild-type pregenomic RNA and basically co-transfect that into cells with something that is a catalytically dead mutant, so this YMHH sort of mutant. I mean, if the wild-type polymerase can function in trans with, sort of complete efficiency, and you put in sort of equimolar amounts of these things, then if you sequence across the active site of the polymerase, you should get equal numbers of reads for both the wild type and the mutant. But it turns out, as shown here, you get very little. So it really demonstrates that, basically the polymerase made by the wild type is not just sort of diffusing around and, sort of copying the sort of the polymerase sort of dead substrate. If you actually introduce these mutations, which basically ablate the sort of pausing function, then what you can see here is that now the sort of wild-type polymerase can actually, sort of work in trans to basically copy some of this polymerase dead mutant, where we've also sort of mutated the pausing. So this sort of dead construct is going to make the polymerase. It's not going to be pausing. So presumably, it could still interact with the packaging signal and sort of prevent the ability of the wild-type polymerase to sort of be incorporated. But if you actually introduce a mutation in that basically blocks the ability of the polymerase to interact with the packaging signal, you can see that we get sort of very high levels of this mutant sort of RNA in trans. So I think this really does show, and this is a nice control for that. If you actually use the wild-type polymerase that can't stall, then it very much sort of restores this cis preference rather than being able to operate sort of on this sort of dead construct. So, I mean, I think it's just sort of fascinating to understand some of these aspects of these viruses. Why have a cis preference? I mean, it probably, and we think it does, affect the replication efficiency of the virus. It makes sense that you wouldn't necessarily want to have your polymerase just go floating off in the cell and not, sort of be a captive audience for sort of making a sort of progeny virus. It could be a selection against defective genomes because it works in cis. If there's a defective, sort of polymerase, it's not going to work. It's not actually going to be packaged into a particle and can spread. Stealth, I mean, I think it could be that, again, this is a reverse transcriptase activity, and one of the activities that you might not want to have as a virus would be a polymerase out in the cytosol making RNA-DNA hybrids and triggering an innate immune response, for example. So maybe this could be used to sort of minimize the generation of those kinds of PABs. And those are some of the things that we're sort of working on. So I think that this RNA launch has really opened up, sort of a lot of opportunities. Deep mutational scanning. I mean, it's amazing that you can get this fitness map across a protein in, basically one step. It's mind-blowing. The initiation of replication using RNA sort of uncouples these cis and trans functions. So, we don't really have to worry about the contribution of these overlapping reading frames. We can really understand sort of more about sort of how these things work without having those sort of confounding things, which are still, I think, very interesting. This is the sort of model. The ribosome pausing tethers the HBV pol to RNA, enforcing this cis-preferential reverse transcription. And I think that I didn't have a chance to, I sort of mentioned this, is that the deep mutational scanning sort of the core region, although it doesn't uncover any information about the importance of particular amino acids in core for function, it basically does sort of reveal sort of nucleotide changes that are associated with this leaky scanning model for basically sort of toning down the sort of level of polymerase that's made from a given pregenomic RNA. So lots of future directions. I think I won't, sort of go through these because I've got something else that I want to show you guys. And that is, sort of switching to hepatitis C. And I think everybody knows that, this was recognized as a post-transfusion cause of hepatitis back in the mid-1970s by Harvey Alter and Fred Prince and others. There was the sort of discovery by Michael Houghton and his colleagues in 1989. And, that really sort of jumping ahead, these are some of the sort of poster children of the drugs that we have today, the viral serine protease, the RNA-dependent RNA polymerase, and then the NS5A protein, which is really, the inhibitors against that are extremely potent. And, we have now treatments that allow us to treat just about everybody and eliminate the virus with 2 to 3 months of treatment. I mean, it's really quite amazing. And so I think for many of us in the room that worked on hepatitis C, sort of having been involved in this process, from sort of the discovery of a new virus to sort of the introduction of the first direct-acting antiviral in 2011, which was basically added unwanted side effects to the already side effect-prone treatment of interferon and ribavirin, to basically an interferon-free therapy. I mean, this was kind of a wonderful, success story to be involved in, and many people, including many people in this room, including this guy over here, sort of made sort of real contributions to that. I think that there are still challenges. I think if we sort of look at this, we still don't have a vaccine for this virus. Implementation, who's infected, affordable treatments. I think advances on the public health front are really quite variable, sort of depending upon the country. And I was struck by this paper, which came from the Center for Disease Analysis Foundation. And basically it shows sort of on the left, where these countries were in 2015 and then 5 years later. And this is in the era of this, sort of direct-acting antiviral therapy. And embarrassingly enough, if you take a look at the USA, which is, let's see, 1, 2, about 7 down here, the actual sort of frequency of HCV positivity is actually going up, sort of not down. And that is in contrast to what we see in Egypt, where they've sort of basically fell 12 places because of their successful program to actually produce these antiviral drugs in the country and have them at a price that they can afford in their public health system. 10% of the Egyptian population was chronically infected with this virus. Now they've managed to eliminate the virus in more than 90% of those infected. And they're probably, sort of on a path to, sort of eradicating this virus in Egypt. So there really are, I think some improvements that need to be made. I mean, the good news with sort of successful HCV treatment is that it does lower the risk of developing liver cancer. As shown here on the left. On the right, the bad news is that there are risk factors sort of associated with, sort of developing liver cancer, untreated HCV and HBV, but cured HCV and suppressed HBV still are at increased risk of developing liver cancer. So that's the bad news, and we don't really understand, why people that are successfully eliminating the virus are still prone to developing liver cancer, what the mechanism of that is. So again, I think we don't have a vaccine. Hopefully here at the IPK, people will be able to make some progress on that with messenger RNA and lipid nanoparticles. And I think on the disease progression front, we really have a lot of gaps in our understanding of, basically how this chronic viral infection leads to progressive fibrosis, cirrhosis, and hepatocellular carcinoma. And also, sort of when this process has occurred, whether it's due to, say, expression of a viral gene or this sort of chronic inflammation that goes on in the liver for decades. So I think one of the things that limited this is really sort of not having an animal model that we could use to study this for hepatitis C. I think as many of you know, the chimpanzee was the animal model for hepatitis C. We spent, and others have spent, a lot of time actually trying to come up with a small animal model. People tried adapting it to mouse cells. I don't think that's really worked. We spent a lot of time trying to understand what are the human factors that sort of restrict the species tropism of HCV. And we managed to sort of make a mouse that could be infected with hepatitis C, but there was no pathology, and it was basically a transient low-level infection. And then I guess the other thing was to actually, just find a natural isolate, a related virus that could be studied. So I'll just sort of go through this relatively quickly because I think how are we doing on time, Sanjay?
You can do whatever you do, but you like, but time is almost there.
Okay, time is almost there. Okay. Uh-oh. Okay, well, we'll go fast. Okay, so I think with next generation sequencing and, basically people, sort of looking sort of more broadly at virus distribution. I had a friend, Ian Lipkin at Columbia, who kept calling me up on the phone and saying that he had discovered a new hepatitis C-related virus. So the first one was in horses, and this is not exactly a small animal model, and certainly not one that we can have on the Rockefeller campus. This is a relatively small campus. We did do some work on that with Cornell and Ithaca. He found actually something in a deer mouse that could establish a chronic infection, a hepatitis virus. And that was exciting. We got the virus. It did establish chronic infections in deer mice, but it turned out that deer mice are so different than laboratory mice that none of the reagents that we knew and loved for working on laboratory mice would work on. We tried to adapt that virus to laboratory mice and failed miserably. There were hepatitis C-related viruses found in bats, in gibbons, but really it turned out that sort of the model that we had been looking for sort of 20 plus years was actually scampering around the streets of New York. And I apologize for those of you that are not rat aficionados here. But so Ian isolated 2 lineages of hepatitis virus from Norway rats in New York. We got a hold of that virus and actually could show that it could be used to experimentally infect laboratory mice, BALB/c mice, C57 black 6 mice. Eva Bellerbeck sort of did this work. The virus in those immunocompetent mice was cleared in 2 to 3 weeks, and that was associated with basically the appearance of CD4 and CD8 T cells sort of in the liver, as shown here. But if you actually transiently depleted CD4 T cells from these animals sort of before infecting them with this virus, we could actually establish chronic infections. And so this was, sort of a model where we had sort of both an acute resolving situation and one that was chronically infected. So the CD4s come back, but they can't eliminate the virus. This is kind of similar to what we saw in the chimp sort of model with hepatitis C. And I won't go through this, but it's actually quite a, sort of a very interesting model where we can begin to, sort of study this. And in terms of the immune reaction, it's still dependent upon this microRNA that aids CB. So they're very similar properties of this small animal model to what we see with people in humans or the early studies in chimpanzees. And, this is potentially a model to test at least platforms for vaccination. But, is it really sort of a model for liver disease? Because, that's, I think, where we were really lacking. So it turned out that there were basically 2 mice sort of in our animal facility that had somehow escaped being sort of sacrificed, and these were sort of discovered in a cage after about 2 years of chronic infection with this Norway rat virus. And as you can see here, sort of the sort of liver on these animals didn't look so good. But, when you've got a 2-year-old mouse, it's kind of like old humans. Sometimes things, don't look so great. And so anyway, Mariana Batista decided to actually test this along with a number of other people sort of in the group. And this is something that we've been working on now for some years. And I won't go through the details here, but basically you do the CD4 depletion, infect the mice with about 10 to the 4th genome equivalents of this Norway rat virus, and then basically have sort of weekly time points where you can measure viremia over time, and then basically terminal time points at 6, 12, and 18 months. This attempt actually sort of coincided with COVID-19, so things were not exactly optimal for sort of analyzing these samples. But this just shows you the viremia in these animals over sort of 18 months. We didn't see, sort of a big difference between males and females in terms of viremia. There was a higher late clearance rate in females compared to males, although, the numbers here are small. And so maybe there are some sex-dependent differences, but that's something that we're looking at in more detail now. If you look at the sort of pathology, and this was read sort of blindly by Neil, a pathologist at NYU, we do see, basically accumulation of fibrosis over time in these animals. And really not, again, a big significant difference between males and females. Progressing, and you can see at 18 months things start to break down because even the control mice are showing sort of some evidence of liver damage. But then it was really surprising in the sense that at these terminal time points, particularly the last one at 18 months, these are the mock-infected mice of the same age, and these are the ones that had chronic NRHD infection. And basically, these tumors were found in sort of essentially all of the male mice, sort of by 18 months, and about half of the female mice is shown here. So there was some difference in tumor volume. I'm not sure that's sort of holding up in sort of our second cohort of these animals. The pathology showed some very similar features to what we see in humans, well-differentiated hepatocyte-like. There are evidence of steatotic hepatocytes that you can see here with the sort of yellow arrows, inflammation, and the presence of these sort of lymphoid aggregates that people were getting quite interested in as to what they might be sort of up to in the liver and also in the sort of liver-associated tumors. Similar sort of transcriptional profiles for sort of basically not tumors, but adjacent sort of non-tumor tissue in both people and these NRHB-infected mice, and we started looking at basically some of the sort of genetics of these tumors as shown here. I won't go into this. So we're actually very excited about this model and sort of what it can be used to understand about the mechanism of carcinogenesis associated with chronic hepatoviral infection. We're looking at the effects of sex and comorbidities, the role of fibrosis in HCC development. These animals do not develop cirrhosis, so it's still, I think, being argued as to whether or not cirrhosis is a required precursor to hepatocellular carcinoma, or it's just sort of a marker of the liver damage that's associated with the development of cancer. It turns out that the other thing that we're working on is actually being able to cure these animals at will. Some of the drugs that have been working for HCV actually do work to some extent with sort of this virus in these animals, but not as well as it does in humans yet, so we're working on that. Okay, so I think I better move ahead. Oh, actually, in terms of host genetics, that's really interesting. So in this model, as I've mentioned, we have to deplete CD4s in order to get chronic infection. Tim Sheehan at UNC has actually looked at the mice that are in the collaborative cross. I don't know if you guys are familiar with this mouse resource, but these are sort of very diverse sort of founder mice that are still inbred, and you can basically infect them with, or perturb them in any way. And if you actually see a difference, you can actually do mouse genetics. And Tim has actually found a collaborative cross mouse, CCO71, where chronic infection is established without perturbing CD4. So you can actually then use that to determine, sort of what are the determinants of chronicity versus resolution of this virus infection. So these are the folks that have been involved in what I've just told you about. Mariana Batista has really been one of the drivers of this. And in HBV, Bill Snyder and Yingpu Yu have really sort of taken the reins of this RNA launch strategy. And I thought I would just sort of take a few slides. I think Takashi is probably going to talk more about, sort of the pandemic. But, these infectious diseases are not going away. And, effective control is really a sort of a global problem, I think. And, we can't be sort of isolated. These viruses can hop on planes and go to different countries. I think we need to have sustained support for cutting-edge infectious disease research, have a balanced portfolio, and have really sort of, very basic as well as sort of applied programs, sort of be prepared and stay prepared, be proactive rather than reactive, and, sort of build local and global infrastructure, I think is really, really important, and foster open communication and cooperation. And I think this is, at least some of these things, I wasn't here this morning, are probably a pretty good match with the IPK vision. And I want to just sort of conclude on something that sort of describes sort of my sort of experience with IPK, a bit of history from sort of 2009 and HCV. And I was invited to sort of come to IPK and give a seminar in 2009, and we had just sort of developed this system for HCV where we could actually monitor in live cell imaging sort of virus infection and spread. So these are basically viral antigens or fusion proteins on the left, and then basically a reporter that upon cleavage by the HCV protease relocalizes to the nucleus on the right. So I presented this here at IPK, and it was basically Mark Windisch, who's no longer here, but I think he was here for a while. Some of you may remember him. Sort of invited me to come here and really got, sort of quite interested in this. And one thing led to another. And basically, two people from my lab, shown here on the left, Chris Joan and Catherine, we call Cat Murray, actually sort of came here, I think just as this space was being finished in 2010. And I have very little evidence that they actually did much work here, because all of the pictures from this time are basically people at restaurants eating a lot. I think here on the lower right, they're at a basketball game. There was also evidence that they were actually playing basketball. I don't know if the court is still here.
Yeah.
And anyway, there was scant evidence that Chris actually was here on, sort of doing some work. And eventually, some years later, we co-published this paper with sort of Mark and the IPK group. And so I just want to sort of, I'll stop there and just sort of wish you a great sort of next 20 years. I think the previous 20 years have been wonderful. And I think this construct where, sort of there is this sort of continuous support so that you don't go through this crazy sort of feast and famine in infectious disease research really can have a huge impact. And I hope it sort of serves as a role model for what some of us should be doing in other countries as well. Thank you.